All right. Hi, everybody. We're going to be moving right along here to part 4 of the digestive system series of lectures, in which we're going to talk about the small and the large intestines as well as the anus. So let's just get into the histology of the small intestine. It's the same basic idea and overall paradigm that you saw with the alimentary canal, all of it. So we have a mucosa, which consists of an epithelium, a lamina propria of areolar connective tissue, a muscularis mucosae. Then you have a submucosa level of areolar connective tissue. Then you have a muscularis of 1, 2, or 3 layers of muscle, which could be smooth or skeletal depending on where you are. And then a serosa or an adventitia, depending on whether you're part of the peritoneum or you're not. So the first part of the small intestine is the duodenum, which has actually lost its serosa and has become retroperitoneal, if you recall from that lecture. So the duodenum will have an adventitia on its outer layer as opposed to a serosa, whereas the jejunum and the ileum, which are the other parts of the small intestine, will not. They will have a serosa because they are part of the peritoneum. Okay, so when we're dealing with the small intestine, the epithelium is a simple columnar epithelium, but there is some diversity in cell types. So there's several different types of cells that you'll see in the small intestine and the epithelium. We have absorptive cells. These are responsible for digesting and absorbing nutrients. We have goblet cells, which I've shown you earlier in the course, whose job is to secrete mucus into the lumen of the small intestine. We have Paneth cells, which secrete lysozyme. Remember, that's the antibiotic that's naturally produced in the human body. They also act as phagocytic cells, so they can digest other materials and provide an immune function. And then there's also enteroendocrine cells, of which there are actually quite a number, that secrete hormones that help regulate the gastrointestinal tract's activity. So like the stomach, you notice that the epithelium deeply curves inwards and penetrates the lamina propria, the areolar connective tissue here. Ultimately, what this creates are epithelial folds, and these epithelial folds are these hairs of epithelia cells projecting into the lumen of the tube, and these are called villi. The lamina propria in the small intestine is areolar connective tissue, but I want to focus on the various vessels that are found there, lymph nodes in particular, those lacteals that we were talking about in previous classes, that MALT, the mucosa-associated lymphoid tissue, that's also very present in this region here. The final layer of the mucosa is this small layer of muscle that I'm highlighting with my cursor right here. That's the muscularis mucosae. That creates small movements and gives characteristic folds to the small intestine, just like it does in the other regions of the gastrointestinal tract. And then underneath that, in this kind of whitish area here, rich with various blood vessels and lymphatic vessels, is the submucosa, which is areolar connective tissue that contains the larger vessels of the cardiovascular and the lymphatic system. And then you get the smaller vessels that run up through these lacteals here in the villi. So there's the lymphatic nodule being isolated. There you can see the muscularis mucosae, and there you can see the submucosa. The muscularis layer has back to 2 levels. So you have an inner circular layer of muscle right here, and then you have an outer longitudinal layer of muscles right here, and you can actually see the myenteric plexuses that are regulating those activities there. Remember, in the stomach, there were actually 3 layers of muscle, but now we've gone back to 2 here. And then finally, when we're talking about the small intestine, we have a serosa in the jejunum and the ileum. If this was the duodenum, however, this would not be a serosa. This would be an adventitia because the duodenum is retroperitoneal. So here's just showing you the various cells of the small intestine. the cells that we're going to spend the vast majority of the time studying are the predominant cells in the villi, which are called the absorptive cells. They have these characteristic folds on their apical surface called microvilli. So just to keep that in perspective, the epithelium is arranged into villi, these long penetrations into the lamina propria. And then on the surfaces of the cells that are in the villi, there are even more villi on their apical surface, microvilli that are actin-based structures. So this is a massive, massive surface area to volume situation here. And this is where most of the digestion in terms of absorption of nutrients is taking place. So again, we have the absorptive cells, which are helping secrete the chemicals to digest the materials in the lumen of the digestive tract. But they're also absorbing it. So the nutrients that are being digested are going to have to pass through these cells and enter into the bloodstream in these lacteals here. Then there's the goblet cells, which are secreting mucus, which is helping to lubricate the system. We've got enteroendocrine cells that are secreting hormones to regulate the activity of the gastrointestinal tract. And then we have these Paneth cells, which are providing an immunological function by secreting these lysozymes to fight off microorganisms, and also phagocytosis to get rid of various debris and pathogens that may be present. So there's a few unique features of the small intestine, which I've already sort of alluded to already. So yes, there is a constant movement of the infoldings of the small intestine. You can see these infoldings right here, just like the stomach. But within there are these permanent circular folds. So you see how there's like a circle made out of this thing right here? All these folds are making this little pocket. There's another one right here. These are these circular arrangements in the intestine. In fact, they can run as long as 10 millimeters long, so they can be quite large. And the purpose of this is to create a little tidal pool, like to cause some spiraling of the intestinal chyme, so that even though the chyme, the liquid that has the dissolved food in it, is progressively moving forward. I mean, peristalsis is happening here to some extent, but the chyme, as it's getting pushed through the small intestine, it's going to get caught in these circular folds, and they're going to whirlpool, and the spiraling will hold it up within the tube for a little bit longer, which will allow for more digestion and more absorption to occur. The key point that I want to make when we talk about these circular folds here is that they're permanent. These aren't constantly waving in and out. These are permanent structures found throughout the length of the small intestine. And here's just looking at the stomach to show you the comparison between the two. So there's no circular folds in the stomach. The rugae are less clear in this particular picture, and you can see there's a lot more folding and villi that are present in the small intestine versus the stomach. And the key point here is the folds in the stomach are non-permanent. They're created by these rhythmic contractions of the muscularis mucosae, of the mucosal layer. If you look within those large circular folds, that's where you start to see the predominance of the villi. So you can start seeing within these circular folds villi protruding from the surface. Again, these are more permanent. The submucosa is actually even part of the villi here, right? So look how the epithelium is actually penetrating not just into the lamina propria, but also into the submucosa here. So again, the purpose of these villi is to increase the surface area to volume ratio of the intestine so that it maximizes the surface area through which absorption and digestion can take place. And if we zoomed in on one of these cells in the villi, we would see that they also have protrusions on their surface. These almost look like cilia, but they're not. These are actin-based projections on the apical surface of these intestinal cells. And these are just little fingers pointing out from the membranes of each of these cells. So hopefully you're getting the idea here. It's infoldings on infoldings on infoldings. Some of these infoldings are permanent, like the circular folds. Others are just kind of waving and moving around. But the key point here is every cell should at least be positioned in a way that it has contact with the intestine. It can contribute to digestion, and it can contribute to absorption. all of the receptors, all of the enzymes are going to be released from these microvilli here. All right, so onto the large intestine. It's shorter than the small intestine, 1.5 meters long. It connects the ileum, which is the distal part of the small intestine, to the anus. Compared to the small intestine, the large intestine is much wider in diameter than the small intestine. There's a little pouch here at the entrance to the intestine, so this is where the small intestine comes in. There's a little pouch here at the entrance to the small intestine. This is called the cecum. The cecum is inferior to the ileum's entrance into the large intestine. This is the entrance of the large intestine is called the ileocecal valve or sphincter. So that controls the movement of material from the small intestine into the cecum here. The inferior cecum has a little projection inferiorly, and that's the appendix, which we've all heard of before. It's ambiguous as to how much the appendix is doing in humans, but it's very important in herbivores, so plant eaters. And in fact, if you were to look at a plant eater's large intestine, you would see that they have an enlarged, a considerably larger appendix than what you'd see in a human or a pig, for that matter. The cecum then merges with the colon. The colon has four parts. There's the ascending colon, which is moving superiorly. The transverse colon, which is moving laterally from the right side of the body to the left. Then the descending colon, which is moving back down inferiorly. And then it curves towards the midline again through the sigmoid colon. So the four components of the colon: the ascending colon, the transverse colon, the descending colon, and the sigmoid colon. There are two significant right-angled turns or 90-degree turns of the colon. These are called flexures. When the ascending colon reaches the liver, it turns right. This is called the hepatic flexure. When the transverse colon reaches the spleen, it turns downwards again. This is called the splenic flexure or the left colic flexure. You may also notice that there are these bulges in each segment of the large intestine. In fact, these bulges sort of make the intestine look segmented. These are called haustra. And you're also going to notice that there is a straight line that's kind of running between those haustra all the way through the ascending, transverse, descending, and sigmoid colon. These are called the teni coli. And these are straight bands of a longitudinal smooth muscle, and they're only contractions. They only do one type of contraction, and those contractions are to actually create the haustra. So these teni coli are creating these bulges that are called the haustra that kind of define the look of the large intestine. And you'll also notice that periodically, as you move up through the teni coli, there are these periodic pouches of fat, these yellow pouches of fat that are running along. These are called omental appendices. Just to look at the histology of the large intestine here, I don't need to beat a dead horse. You should be pretty familiar with the mucosa, submucosa, muscularis, serosa components here. The epithelium, because the material in the large intestine is still chyme, so it's just a dissolved materials in water. We only need a simple columnar epithelium here. There's no projection, so there's no need for villi. There's no need for microvilli here because there's very little secretion and absorption that's taking place in the large intestine. The infoldings that you see in which the epithelium penetrates into the lamina propria are called intestinal glands, which are responsible for secreting various materials into the large intestine. But the key point here when you consider the large intestine is it's nothing compared to the small intestine with respect to those massive infoldings. The large intestine doesn't seem to be dealing with the same sort of surface area to volume ratio that the small intestine had to deal with. The large intestine is much less infolded. At the end of the GI tract is the rectum. It's 20 centimeters long. Its final section is called the anal canal. The anus is the opening of the anal canal to the outside, and whether or not the anal canal is open or closed is dependent upon two sphincter muscles. There's an internal anal sphincter, which is made up of smooth muscle and therefore is involuntary. And we have an external anal sphincter, which is right at the distal end, which is voluntary and hence made up of skeletal muscle. In terms of tissue, you would expect the exact same types of tissue: mucosa, submucosa, muscularis, adventitia, or serosa, but the epithelium in the anus is going to transition again from being simple columnar to being stratified squamous. And the reason for that should be clear is because as the chyme is moving through the large intestine, the water is being absorbed in the large intestine, and so the feces is starting to form. It's starting to become more solid. And again, whenever you have solid material, the tube that's containing that solid material is going to need to have a stratified squamous epithelium. Okay, so that ends our short and brief tour through the digestive tract. We'll talk a little bit more in the coming lectures on some of the physiological processes that happen as we go through this tube. Okay, see you soon.